In the heart of a major European horticultural and seed production area in the region Pays-de-la-Loire, INRA, Agrocampus West and the University of Angers joined forces in January 2012 to create a large plant science laboratory in Angers.
The Research Institute of Horticulture and Seeds groups together 240 researchers and PhD students, specialized in physiology, ecophysiology, genetics, pathology and modeling.
It forms a major part of the SFR, Quality and Health of Plants, and is member of the competitiveness cluster, Vegepolis.
The teams of the pole Seeds and Stress develop research projects using Medicago as model to understand the acquisition of stress tolerance, maturation processes, survival of seeds in dry conditions and germination quality.
They have identified the role of LEA and HSP protein within the seed proteome and the adaptation of mitochondria in extreme conditions.
The role of nutrients such as nitrogen during the post-germinative phase under water stress, and of antioxidizing enzymes during germination.
They characterize QTL, clone genes of interest, and develop transcriptomic approach, taking advantage of nucleic acid analysis platform of the SFR to identify key regulators and gene network which control this trait,
and predictive markers of physiological quality. The imaging platform of the SFR, hosted by INRA, provides facilities for cytological and histological studies. Furthermore, the SFR and the GEDES led the phenotype project, which resulted in the creation of a phenotyping platform to characterize precisely the germination of large sets of seeds by imaging or seed quality by radiography.
The phytopathologists of the MRC's team study the evolutionary history of plant-associated bacteria.
With the Xanthomonas genus as a model, of which all plant pathogenic species are transmitted by seeds and young plants,
they study the genetic structure of microbial communities,
their ability to be transmitted to and by seeds,
and they characterize host adaptation mechanisms.
The French collection of plant-associated bacteria, recognized worldwide among the plant pathologists,
is a strong support to these projects, to study host-pathogen interactions, to identify the molecular determinants of emerging pathogens, and in order to provide new detection tools and control methods.
The researchers of the Fungisem team study the adaptation of seed-borne fungi to environmental and host constraints,
in particular species from the Alternaria genus, by phenomic and transcriptomic approaches,
to identify signaling pathways which bypass plant defenses, such as the hog and the CWY cascades, and to discover new control methods.
Both teams are now developing an ambitious metagenomic project on plant-associated microbial communities. France is one of the world leaders in carrot seed production.
But the genetic knowledge of this species is still poor.
That's why the team working on carrots studies the genetic diversity and the genetic structure of carrot populations,
taking advantage of a large genetic resource collection, which is also a worldwide reference and a strong support for selection programs.
The team is part of an international carrot genome sequencing project.
Researchers are identifying major QTLs and studying the molecular mechanism controlling aspects of fruit quality related to carotenoids.
An interaction with the main carrot pathogens such as Alternariados.
INRAE-Angers has a long research background on palm fruits and has developed large genetic resources used in the selection programs in collaboration with our private partners to create new high-quality and resistant cultivars.
This provided the opportunity to participate in the apple genome sequencing project published in 2010.
The main research and selection goals are fruit quality based on sensorial analysis, biophysics and biochemistry.
And sustainable resistance to pathogens, particularly apple scab.
A team of phytopathologists study the genetic diversity of the fungus by genome sequencing of numerous strains,
and study its evolutionary capacities in relation to its host to provide strategies for the sustainable management of the adaptative dynamics of the pathogens,
by modeling of disease epidemiology and new management methods for orchids.
Other researchers use biotechnologies to understand the response of pears and apples to fire blight,
and to decipher the defense mechanisms which are mobilized, in particular those induced by natural defense stimulators.
This led to the creation of a service platform with Vegepolis Innovation.
A similar approach is developed on susceptibility to aphids.
For all these traits, QTLs have been identified by genetic mapping,
and some major QTLs are being cloned.
To decipher the genetic control and molecular determinants of these traits, functional genomic studies based on transcriptomics are performed by these teams.
We are on the way to genomic selection thanks to the large European project Fruit Breedomics, coordinated by the laboratory. The Angers laboratory is working on ornamentals with rose as a model.
Researchers are studying plant architecture focusing on budding using an integrative strategy,
taking into account environmental effects, such as water supply and light,
as well as sugar metabolism and related genes, to develop comprehensive structure-function models.
Plant architecture is digitalized and plant growth is modeled.
The laboratory has large genetic resources which support genetic diversity studies
and genetic studies on ornamental traits such as recurrent blooming.
The cloned genes are functionally validated by biotechnologies.
The researchers also work on disease resistance in rose in collaboration with private partners.
In parallel, there are several other projects also involving private partners
to transfer the fundamental knowledge to other ornamentals. Thanks to his experience in rose genetics, the laboratory today co-leads the rose genome sequencing project.